Hello, thank you for the opportunity to give you a glimpse of my work. My name is Anya and I'm a postdoc in the Sabatini lab. And I'm gonna take you through the ages and tell you about our exploration of mitochondrial structure and function. The image that is in the gallery was taken in collaboration with Peggy Bisher and David Mankus from the NanoCore at the Koch Institute. So we live in a rapidly aging world, and across the globe, you can see that the average age of societies have been increasing. This has been happening over time, and it's projected that this tendency is gonna continue in the future, so that in 2050, we will have 2 billion people who are aged 65 and over. And this produces a number of challenges for societies across the globe. For example, aging is one of the factors, risk factors for many age-related diseases. And so there's a lot of interest in the biomedical community in understanding the causes and mechanisms that underlie aging, with the hope that in the future we will be able to better address diseases of old age. There's a number of molecular features of aging cells that are quite distinct and different from those of young cells. And these vary from differences in communication between cells, the regulated nutrient sensing, or differences in genomic stability. But one interesting fact is also that the compartments within cells age as well. And the mitochondrium is one of the organelles that is known to age and undergo most dramatic changes. So what are mitochondria? Mitochondria are compartments within cells, and here they're shown in red. And you may remember that they are the powerhouses of the cell because they provide energy for lots of different activities that cells perform. However, mitochondria are much more than that. They're important for a number of different metabolic pathways. They are key hubs for cellular signaling, and they can even decide cellular fate. As I said, they are one of the organelles that are known to undergo very dramatic changes in aging. And while there is lots of very interesting and beautiful literature out there on characteristics of aged mitochondria, what has been missing is a very comprehensive and unbiased analysis of aged and young mitochondria. And this has not been possible before because we have lacked tools to do such a big analysis. However, in our lab, we have developed new biochemical approaches that allow us to rapidly isolate mitochondria from young and aged tissues. And these approaches are compatible with a number of downstream analysis platforms, which allow us to profile metabolites and proteins and really zoom into very detailed changes in composition of mitochondria as we age. We hope to combine this biochemical analysis with powerful imaging approaches to look at structure, ultrastructure, numbers, and sizes of mitochondria through the ages. And this image represents one of the images that we have taken across the tissues and through the ages. It is an image taken in the liver, and each of these colored blocks represents a single separate cell. You can see within each of the cell, you can see these membrane-bound compartments, and these are the mitochondria. And we are currently performing analyses of the young and aged mitochondria in different tissues and comparing their size, structure, and number. And we hope to combine this with our biochemical analysis to form novel hypotheses about what happens in aging. And with that, I would like to thank David Sabatini, who is the fearless leader in our lab, for his support and encouragement to work on this project. I would like to thank the Peterson Nanotechnology Materials Core Facility, Peggy, David, and Abigail, without whom the electron microscopy image and the whole series of the images we've taken would never be possible. And I would like to thank all the members of the Sabatini lab, as well as the Metabolomics Core, for creating a wonderful environment to work and do science. And thank you all for listening.